So I'm Jared. I work for Dr. Hans Lockmuller over in Canada at the CHEO Research Institute. And I work there as a research assistant, doing all sorts of work with zebrafish and developing different genetic models for neuromuscular diseases. So, as we all know, as Rudiger so elegantly explained, there's a lot of things that have been wrong with the mouse models that have been trying to develop, whether it's Dr. Ishino's lab or Dr. Rosenbaum's lab. A lot of work has been put into trying to develop these models to really no avail. So that's where zebrafish come in. And if you're a patient, you've never really heard of studies being done in zebrafish, they're a really great model. So why should we use zebrafish in research? Well, zebrafish and humans share about 70% of all human genes. They have 70% of those genes in common. And about 80% of known human disease genes are also found in zebrafish. So we can do knockout studies in zebrafish for many known human diseases, including GNE. They have a lot of other benefits. They're very transparent during their development. So you can look at internal organ development and get a lot of measurements without invasive procedures. Now they're very highly fecund, so they have very large clutches of eggs, anywhere from 200 to 500 eggs per clutch. So this also gives your experiments very high statistical power when you run them. Additionally, they're very easy to maintain and genetically manipulate because of external fertilization. So you can basically take a zebrafish egg once it's been laid and do injections of something like a morpholino right at the one cell stage and create knockdown models. So we're kind of building on the backs of giants. So everyone has now heard of Dr. Stella Rosenbaum. So she was actually the first one to develop one of these zebrafish models in her lab with her postdoc, Alan Dea. And so if you don't know what a morpholino is, it is an antisense oligonucleotide that permanently binds to its target transcript. So in this case, they developed a morpholino sequence to target the E3, I3, or the exon 3, intron 3 border to block splicing and cause nonsense mediated decay of the GNE transcript. So you get a knockdown in GNE protein expression. And the phenotype was pretty severe. The survivability of these fish was around 30%. And then the fish that did survive kind of had a distributed phenotype from normal and mild fish to intermediate and to severe. So you can see an obvious reduction in size, tail curvature, pulmonary edema, a buildup of fluid in the pericardial space, and then they also get very, very small. And as controls for all morpholino experiments, you need to do rescues with injected mRNA of a healthy version of the transcript. And they were actually able to rescue almost completely the phenotype upon wild type GNE mRNA injection into these morpholino fish. So that was really great to see. They also had major muscle fiber defects, disorganization of slow muscle fibers. You can see there are large gaps between the fibers and they're very wavy as compared to the controls, which are tightly packed and organized. So the great thing is that reproducibility of this model is awesome. We have been doing injections with this same morpholino in fish and been seeing roughly the same results. So distribution of the phenotype, roughly the same survivability in these morpholino fish. And in addition, so we did injections into the wild type strain, just like Stella did, but we also did injections into an optogenetic line of zebrafish. And again, on a different genetic background, saw roughly the same phenotype development. Now, the interesting about this optogenetic line, if you don't know what optogenetics are, basically these fish express a ion channel, channel rhodopsin, in their motor neurons that respond to blue light, and they open and cause the motor neuron to send a signal and cause muscle contraction. So we can stimulate muscle contraction via blue light stimulus. And that helps with kind of defining any sort of locomotor issues that the fish may have when we're injecting different morpholinos. So when we did these injections, we also run different types of behavioral assays. This one is called the light dark challenge. So it's kind of opposite as to what you might think. But in the light, in just plain white light, zebrafish larvae, they don't like to move. But as soon as you switch the light off, they kind of gain some confidence and start moving around. So you can see nicely that the wild type uninjected fish and control injected fish, they move pretty nicely, whereas the wild type or channel rhodopsin injector, the GNE injected fish, do not move nearly as much as their control counterparts. So this is really great because we know that it's reducible not only between labs, but across different genetic lines, and we see major locomotor phenotype. But there are pros and cons to the morpholino model. The pros being this model has a very quick phenotype. You start seeing differences at day one of development. You can also do these experiments very fast. 5 days is the length of an experiment, whereas mouse models you can go for weeks. Obviously, it's relatively cheap. You kind of just got to keep the fish in water and they're good to go. And then, as I said before, you can inject many different lines with this morpholino and see the same phenotype. So you can also go into different reporter lines to look at different specific things. But one of the main concerns with any morpholino model is the off-target effects of the morpholino itself. There are many control experiments involved in determining the specificity of the phenotype to the injection of the morpholino that are done in the background. That I'll point you to Stella's paper for that. But the other thing is that it's just a transient knockdown. So as the fish divides and grows and expresses GNE, the amount of morpholino is finite. So when cells divide, the morpholino is divided in half and is used up as the GNE transcripts are expressed. So you kind of get fading knockdown over time, which is not as good for observing later time points, such as with progressive diseases like GNE or GNE myopathy. So Stella again, actually this time her postdoc who moved on from her lab, they actually developed the first CRISPR knockout model of zebrafish GNE. So they found and targeted with their CRISPR knockout exon 3 and induced frameshift mutations that induced premature stop codons in the GNE gene and successful knockout of the GNE protein. Now, what was interesting with these fish is that they had a progressive phenotype. So it developed over time, starting at about 6 days old. The first thing they kind of noticed was this one of the first things they noticed was this development of a deflated swim bladder. So for those of you who don't know, zebrafish need to actively go to the surface of the water, gulp air to inflate their swim bladder, and that keeps them suspended properly in the water column. But knockout GNE fish, they had slowly deflating swim bladders over time, which isn't conducive to good swimming. They also saw a progressive decrease in skeletal muscle fiber integrity. So this right here is a picture of some zebrafish skeletal muscle using birefringence imaging, uses polarized light to image healthy muscle tissue in white. And then you see faded and unorganized skeletal muscle in the knockouts over time. So at 7 days old, you see the start, 8 days, and it gets worse. So this is really good in terms of a phenotype, as it's more capitulative of the human disease. They also attempted sialic acid treatment in these fish, but unfortunately there was no differences seen, and the fish still died around 10 days old. So kind of at the same time, before their paper was published, Dr. Emily O'Connor, who was part of our lab over at CHEO and has since moved on, was actually part of this consortium and did the initial guide RNA injections into zebrafish to generate our own knockout model of GNE. So she initially generated crispins, which in the development of a CRISPR knockout model, basically have a bunch of random mutations that are induced by that injection of the guide RNA in the Cas9, which is then randomly repaired. Different amounts of nucleotides are inserted or taken away to then repair the DNA break caused by the Cas9. And those deletions are what we were looking at, and what we hope to be in the germline to then create founder fish and build up a colony of these fish. But you can also look at the crispins, although they have a mosaic knockdown and is a result of many different types of mutations. You can still see that it has significantly reduced survival, which is a good thing in our case. She also noted some locomotor and morphological changes, which was also good news for us. So I went and genotyped a whole bunch of different fish, found that we had all of these mutations. Anything that is divisible by 3 here is an in-frame mutation that will not cause any sort of, or were found to not cause any sort of premature stop codon, and you still get expression of GNE. But the 1 base pair and the 2 base pair, which is what we decided to grow up, does induce a frame shift up here and a stop codon to stop expression of GNE. So we wanted to validate our data against the other CRISPR knockout model, and we found that it was actually very consistent. So in terms of the deflated swim bladders proportion of the population, you saw a really large increase in the mutants' deflated swim bladders. So about 30%, 25% of those fish had deflated swim bladders as compared to their wild type and heterozygous clutch mates. And then we also saw the development of a tail bend, which from starting at day 5, day 6, all the way until day 14, and then you see that big drop in survival, kind of starting around day 12 there. And then we also saw progressive locomotor dysfunction. So this is the 5-day-old light dark challenge. You see that the mutants are swimming fine, and then at day 7, you see that they're swimming much less than their clutch mates. So this is all really great. And what I'm trying to tell you is that we have animal models to test drugs in. So we also know that these are like the only real reproducible in vivo models of GNE myopathy. So we have a lot of future work to do. We got to test all these prodrugs, specifically PGD5, and we got to Our plan is to optimize the dose in the morphin fish, which are very quick experiments, and then do final experiments with an optimized dose in the knockout model. And then, so thanks for everyone for giving me the opportunity to talk here. Thank you to Michaela for organizing this, and we can all go have a drink now.
Thank you very much. It's a perfect end to this busy day.